All right, so in 7.2, we're going to talk about mitosis. And just a reminder, mitosis is the process by which a cell replicates its chromosomes and then separates them into two identical daughter cells. So if you look at the picture over here on the right, this was a slide earlier from 7.1 when we talked about the cell cycle. In here, you have just a regular old cell, right? It looks pretty normal. This is an animal cell. Once it heads to prophase, you can see that there are four individual colors in there, okay? Those are chromosomes, like we talked about on the previous slide, that are actually in the form of sister chromatids, okay? And you'll know what I'm talking about here in a second. But as we go through prophase and then into metaphase, you can see that they line up down the middle. Anaphase, they come apart. And in telophase, you can see that there are still four chromosomes in this cell kind of up above, and then four chromosomes down in the cell below. And so what we're going to do is we're just going to go step by step through prophase, metaphase, anaphase, and telophase. Okay, and so in mitosis, I'm going to use the same picture throughout all the phases. It's from the same website, so it's consistent. But DNA is going to be coiled into chromosomes. So as you can see here, kind of like what we talked about previously, DNA, which gets wrapped around these histone proteins, become nucleosomes, and when we get a bunch of nucleosomes, it forms this structure, and that's what this is here. Okay, these are sister chromatids, also otherwise known as duplicated chromosomes because there's two copies. It's still one chromosome. So if you're probably thinking, what the heck, how do I know how many chromosomes there are? Count the number of centromeres. Okay, here it says that the centromeres appear. That's the circles in the middle. So there's 1, 2, 3, and 4. Okay, and so in prophase, here's what happens. You get the nucleus starts to disappear. So that's this purple spot right here, this kind of pink purple spot. You can see that that circle is disappearing. The centromeres appear, those circles in the middle of the chromosomes. And then the centrioles appear in animals, not in plants. The centrioles are these green pieces here. Okay, so they are going to appear, and then they're going to start to kind of head to opposite directions of the cell. In metaphase, this is really easy to remember. What I tell students all the time is metaphase middle. As you can see, where do the chromosomes line up? They line up right down the middle. Okay, so the chromatids move right down the middle. Spindle fibers, which are these kind of purple lines that are extending from the centrioles, they attach to the centromere. So that center of those chromatids, that's where they're going to attach. Okay, so metaphase middle, that's the big piece that I need you to remember. In anaphase, I always like to say anaphase apart. Okay, so what we're doing is we're separating those sister chromatids. Okay, so if you look over here, okay, if you're wondering, okay, how many chromosomes are there that are going to head this way? Well, count the centromeres. There's 1, 2, 3, and 4. There's 1, 2, 3, and 4. So the question is, how many chromosomes are going to be in each individual daughter cell? The same than what we started with. Okay, so they're now separating to opposite poles. So those centrioles with those spindle fibers, in class I like to describe them as hands grabbing onto something. So they're like hands extending, grabbing onto the centromere of the chromosomes to pull them apart. In telophase, again, you can see they're really starting to separate. The nucleus is going to start to reappear. That nuclear envelope, just the very outside of the nucleus, is going to reappear. And again, you can see that there are 2 blue chromosomes and 2 red chromosomes. 2 blue chromosomes and 2 red chromosomes. Okay, those spindle fibers are going to disappear. The chromosomes are going to relax and kind of head back into chromatin, a less dense version of DNA than a chromosome. And what we're going to see is we're going to start to see this cleavage furrow form. We're going to talk about that here in just a second with cytokinesis. So lastly, in cytokinesis, this is the final stage of cell division where the cytoplasm, so the liquid space in between the nucleus and the cell membrane, that's going to separate. And what we're going to have is we're going to have 2 identical daughter cells. So you can see this picture isn't my favorite, but you can see the centromeres here, right? You can still kind of see them. They kept them in there to let you know that there are 4 chromosomes on this side and 4 chromosomes on this side. Okay? And when we're talking about that cleavage furrow, that's the pinching. So it'd be like if I take my 2 fingers with these 2 cells and pinch on the sides. That's kind of what the cleavage furrow looks like. Okay? And in plant cells, it's a little bit different. They form a cell plate. Imagine like it's a dotted line where the cell knows to take a pair of scissors and cut right down the middle. Okay? So it's very similar. Animal cells, they form that cleavage furrow like we talked about a little bit in telophase. In plant cells, they have a dotted line that shows you where the cell plate is. So those are the 4 steps of mitosis, where we have prophase, metaphase, anaphase, telophase, and then the official, excuse me, I'm battling a cold right now. And the official split of the 2 cells, that would be cytokinesis. And so we're going to pick it up with meiosis in the next video.